Okay. So good morning, everybody. Okay. Good morning. Today we discuss the sixth paper, that is the medicinal botany and embryology. Embryology. So in this sixth paper, we have the 5 units are there. The 5 blocks are there, sorry. So in this one, here 5 blocks are there. So in block 1, here consists of 4 units are there. So in first unit, the role of the medicinal plant in medicine, origin and development of the different system of medicine. So what is the main role in the medicine? The plants are to play an important role. What is the important role in the medicine? And origin and development of the different system of medicine. Different system of medicine means Siddha, Allopathy, Unani, and Homeopathy. So here 4 systems are there. So these systems we are discussed that one. And the next one is the next unit is the general accounts of phytochemistry of medicinal plants. So second unit, the general account of phytochemistry of medicinal plants. So we discuss the what are the chemical constants are there. Different types of the chemical constants are there in the medicinal plant. And third unit, the morphology, active principle, and the medicinal value. So what are the active principles in the specific medicinal plants we are discussed, and medicinally their values also. Each plant we have the active principle is there. The special compound are there. Special type of the compounds are present in the in that medicinal plant, which are the medicinally more valuable. And the fourth unit is the morphology and active principles of another medicinal plants are there. This is also the unit third and fourth. So both are the discuss the active principles and the morphology of their chemical content and the medicinal values. We are discussed that one. The second block here. The second block okay. So here the second block again consists of 4 units are there. In this medicinal plants unit 2, that is the block 2 here. In block 2, here fifth unit, the cultivation of medicinal plants. The fifth one, here we discuss the cultivation of medicinal plants. Some important medicinal plants are there, we have to cultivate. So, Vitamnia like that, for different medicinal plants are there. How to cultivate in that medicinal plant. So, unit 6, that is the pharmacognosy and adulteration of plant drugs. Pharmacognosy and adulteration of plant drugs. So here we discuss the pharmacognosy. What is the pharmacognosy? And another subject is the adulteration of plant drugs. Okay. So here the different types of adulterated are there. We are discussed that one. And the 7th unit is the ethnobotany. Ethnobotany is history, scope, and its importance. So what is the ethnobotany? We described that one. the definition of the ethnobotany, what are the history, their history, and what are the scope and their importance. So nowadays, the study about the medicinal plants, that is the traditionally, that is the ethnobotany, we discussed that one in 7th unit. In the 8th unit, the conservation of the medicinal plant. The 8th unit is the conservation of medicinal plant. How to conserve, how to protect these valuable medicinal plants. We are to discuss in this unit. So most of the all plants are medicinally more valuable. So that's why we should protect their life. We should protect or to cultivate our conservation. We are conserve that one, inside to conservation and outside to conservation, to protect these valuable medicinal plants. And here black third, that is the embryology of angiosperms. From here, third and fourth, fifth, these three blocks consist of embryology, belongs to the embryology. So third block is consist of 4 units are there. So in this one, the structure, ninth unit. So here, ninth unit is the structure of anther and development of the male gametophyte. Structure of anther and the development of the male gametophyte. So we discussed that one. The 10th one, the structure of ovule and the development of male gametophyte. The structure of ovule and the development of the male gametophyte, we are discussing in this block. And the 11th unit is the fertilization. 11th one is the fertilization. We discussed the importance of fertilization and what is the fertilization. The 12th one here, the sexual incompatibility. The 12th one is the sexual incompatibility. So some adaptations are there in the self reproduction. So here some adaptations are there in the sexual incompatibility. We discussed in the 12th unit. And the black fourth, this one also belongs to the embryology. Here we discuss in 13th unit, the development of endosperm. So after fertilization, the endosperm is there. That endosperm is what is the development stages? We discuss in this one. And the 14th unit here, the development of embryo. The development of embryo, discuss that one. The 15th one here, apomixis. The 15th one, apomixis, discuss one. And the 16th unit is the polyembryony. The 16th unit is the polyembryony. Okay. Then the block 5th discuss the 17th unit, that is the parthenocarpy. 17th unit is the parthenocarpy. And the 18th one is the experimental embryology. Experimental embryology. And 19th one is the applications of embryology in taxonomy, as well as the agriculture and horticulture. So how they utilize in the taxonomy, agriculture and horticulture are the main branch of the embryology. What are the applications are there? To identify the species. So as well as in the agriculture and horticulture. And the 10th unit is the principles of and applications of field knowledge. This is a very important topic, is the very interesting. The curiosity to develop, to discuss about the field knowledge. Very important topic, this one, and very interesting also. The principles and applications of field knowledge. In this one, we discuss the one by one. Okay. So here, the first of all, we discuss from the embryology topics we are discussed. Okay, so embryology we discussed that one. So from here, the anther structure. So here discuss the structure of the anther and male reproductive development, means the male gametophyte development we discussed in this. Today we discuss that. So first of all, the anther initiation. So here, so the anther, what is the here? The dynamic of the anther development and the differentiation depending on the cooperative interaction between the cell population. So the anther normally consists of two lobes are there. Okay, so any anther here having the two lobes are there. So again, that each that one is two elongated microsporangia or pollen sacs. That one we call as a the two lobe again consist of two sacs are there. That is the pollen sacs, pollen chambers are there. Each anther is having the two lobes are there. Again, each lobe is consist of two pollen sacs. So the anther lobes are fused together by the connective tissue. That is connected. Connective tissues are there. It is a very sterile tissue, parenchyma tissue. The filament connect the anther to the receptacle in the flower. You know that the filaments are connect the anther with the receptacle. A differentiated anther has several highly specialized cells and tissues that are the responsible for carrying the out of non-productive functions and the reproductive functions are there. You know that one. So here the within the anther here different types of the tissues are there. That is the reproductive functional tissues are there and the non-reproductive functional tissues are there. So here the non-reproductive tissues include the epidermis, endothelium, tapetum, and the circular clusters, connective stomium, and the vascular bundles are there. These are not involved in the reproductive function. So here, so each of these tissues and cell types are carry the a special functions are there. Each group of the cells is involved in the different functions. So in addition to these diploid sporophyte tissues, the anther also contains the haploid microspores that fill the pollen sacs and differentiation into the pollen grain. So here, the different types of the tissues involved in the major functions are there. That is the connective tissue. So connective tissues, which are the joint, the anther and theca, these are the together to connect the anther to the filament to provide the structure and support the and morphology. Connective tissue to join the anther to the filament. And the circular cell cluster, which are involved in the dehiscence of the anther. Another one is the endothelium. Okay, so these endothelium to the major to give the structure and support and dehiscence also. The epidermis, which is give a structure and support and prevent the water loss and some gas exchanges are there. Epidermis also to protect the anther. The microspores, the pollen grains and sperm cells are developed. And another one is the middle layers are there. So these middle layers are structure and support and simultaneously dehiscence the anther. Special types of cells are there, which is called as a stomium. These stomium cells also involved in the dehiscence of the anther. So another one is the tapetum. So the tapetum is a specialized tissue. Specialized tissues are there, which is mainly involved in nutrition for the pollen development. So development of the pollen wall components and nutrition and some enzymes are developed for the microspore. And another one is the vascular bundles. These are the connecting tissue between the anther, filament, and flower. So this is interconnected. Okay. So the anther is the first appearance as a cylindrical structure composed of the mass of the meristematic cell. Okay, which consists of three layers. Three germ layers are there, which are designated as the L1, L2, and L3. Okay, so L1, L2, and L3. So the main layer one. The layer one here. The layer one we call as a, that is the made up of the, which is epidermis. It consists of epidermis. Include the epidermis is the stomium is present, and there are the senescence and stomium involved in the dehiscence. And the layer two, that is the layer two is the developed from the archisporial cells. The archisporial cells are developed. So these archisporial cells again to develop the sporogenous tissue. And another one is the primary parental parietal cells are the sporogenous tissues to develop the microspore mother cells. After microspore mother cells undergoes to the meiosis to form the tetrad pollen. So sporogenous tissue to provide the mother cells of the microspore, that is the having the diploid condition is there. The spore mother cell is having the diploid condition, and it is undergoes to the reductive division to produce the spore mother cell. So after they can develop the four tetrads, which are the separated, and to develop the microspore and to release the pollen grain. So these archisporial again the secondary, that is the to develop as the primary parietal cells. Primary parietal cells, again the primary parietal cells to give the secondary parietal cells. From this one to produce the endothecium. The endothecium is the non-reproductive organ which is involved in the development of the anther. The endothecium again they produce the wall which is involved in the dehiscence of the anther wall. Again, these secondary parietal cells again then develop as the middle layers. The middle wall the outer tapetum is also developed. Okay. So in the third layer, layer 3, which are the connective tissues and vascular bundles. The third type is the connective tissues and vascular bundles are there. In this one, we can develop the intertapetum and circular, that is the cell cluster, circular cell cluster. They are the degenerated and senescence. Okay, so this is the different types of the cell layers are there involved in the. So in most cases, the individual tissues and cell types are derived from the single germ layer. For the instruction of layer 1 is gives rise to the epidermis and the stomach. We already discussed that one. And the layer 2 is gives rise to the archosporial cells and microspore mother cells, endothelium and middle layers also. Okay. So, we show the diagram here. This is the so this is the primordium. This is the to develop a anther. So these archosporial cells are designated to differentiate it into microsporangia and their surrounding tapetum and endothelium tissue. So array these simultaneously in each corner of the anther primordium, while the vascular tissues is differentiated with the center of the another primordium and establish a connection with the phlegm. So thus the specific regions and terrestrial, these are the territorious or the established early in another development between which is unique and histo differentiation even occur in the crisis from logical order. So we discuss the PS of the anther. So here PS of the anther in the anther is consist of the epidermis, endodermis, middle layers, and tapetum. In each anther here, these four parts are there: epidermis, endothelium, middle walls, and the tapetum. So epidermis is a single layer. Okay. So epidermis is a single layer. Like the structures. So here, like this is there. So Here, the epidermis is a single layer, which is the protective layer. The epidermis we call as a protective layer, and this one is the single layer sheath of the anther. It is divides the anticlinally and size to give the space to the enlarging and internal tissues of the anther. As a consequence, they are undergoes to considerable strengthening in the surface area, strengthening in the surface area. It provides the structural integrity to the anther and assistance to gaseous diffusion and to prevent the moisture loss and the dehiscence of the anther lobe. All cells are rectangular cells are there. Is also involved in the dehiscence of the anther. The next layer is the endothelium. So, in the below the epidermis, the layer is the endothelium. Below the epidermis, the parietal cells located below the epidermis is called the endothelium. It attains the maximum development before the dehiscence of the anther. Okay. The cells are uh, radially elongated and decorated with the fibrous bands are there. Fibrous bands are there in the endothelium cell. So these endothelium mainly to involve the dehiscence of the anther. These fibrous bands are there, thickness are there. So that is the so these that is the run upward from the inner tangential wall and ending the near the outer wall of the each cell as an incomplete ring. The outer tangential walls are remain thin. Okay. That is the endothelium is associated with the high proportion of the cellulose, alpha cellulose, and the small amount of the lignin, the maturity level. The main composed of the alpha cellulose. These specialized nature of the endothelium together with the stomium, which is help in the basis of the endothelium is include the some stomiums are there, that is the exchange of the gases. Okay. So third layer is the third portion is the middle layers. So these middle layers are usually ephemeral and became flattened and crushed by the early meiosis in the pollen mother cells. So these layers are persistent in the ranunculus and the lilium family members. So these layers are adjacent to the endothelium, may even develop fibrous thickening. In a few instances, it also serves to the store starch that will be later mobilized to the development of the pollen. This is also on the layer, also having the endothelium that help in the development of the pollen grain. And the last one, innermost layer of the anther that we call as the tapetum. The tapetum is the innermost layer of the anther wall and usually derived from this is the parietal layer, parietal cells. So it may have a dimorphic origin in a few species within the Alatica, and the inner tapetum consists of the large cells that develop from the connective. So, outer tapetum, the smaller cells, which is derived from the parietal layer. Further, the tapetum is also origin from the peripheral cells of the sporogenous tissues. The Maheshwari, which are identified in 1950, and these believe in the parietal origin of the tapetum as the obligated. Okay, so the tapetum is surrounds as a sporogenous tissue and contains the maximum development when the microspores are in the tetrad state. So after which they go in the decline, that is the result in the collapse of the cell. So what I'm here. So these tapetum to act as a sporogenous tissue and to give the nutrition value and they surround the sporogenous. So this is generally a single layer, but in many divide and became the biseriate cells and in the Tacoma and Mongolia. So some multiseriate cells also there. Majorly, the tapetum is a single layer. Some members in the biseriate and some multiseriate layers are more than two layers. So these tapetal cells are a specialized cells are there. A special character is present in these cells are. They are the distinctly elongated and always ephemeral. So these tapetum cells, the rhizoplasm, this means the cytoplasm is rich in ribosomes, mitochondria, endoplasmic reticulum. So number of vesicles are there and they are the active organelles. So include the cytoplasm, rich ribosomes. Plenty of the ribosomes are there, mitochondria. All the organelles are very active. And another character is the cells may be multinucleated. This is a special feature in these tapetum cells. Multinucleate cells are there. And these are the irregular mitotic divisions and the nuclear features. You can see the irregular mitotic division is occurring. They are characterized by the rapid and um, intense activity with the degeneration of the cytoplasm. So, the distinct feature of the tapetal cells is the Golgi body complex, which are the make the essential portion their ultrastructure of the pituitary. So the secretory vesicles that leads toward the side facing of the anther locule and the small locule containing the lipophilic substances are there. So here, so some nucleus here. So what is the functions here? Since these predominantly made the chloropolenin, what is the main some functions are there? These tapetum cells, the development of the microspores. These tapetal cells are predominantly involved in the sporophyllin, development of the sporophyllin. And it may resist to the free passing of the material into the outer and the spore mass. And these perpetual members is considered from the impermeable culture sac, enclosing the young spores and the tapetal plasmodium during the period of the sexine growth. In some members, the family members, that is, the family members of the Cyanopoideae and Ascariaceae, the tapetal members hold in the pollen together to aggregate the essence, their collective dispersion. What are the Ascariaceae members? They can form the pollinia, a group and or a mass of the pollinia together, and they are to dehiscence. That is the mass of the pollinia. That is the merely the anther development. Okay, so here the next one is the here we discuss the okay male gametophyte development. Next one we discuss the microsporogenesis and no microspores, that is the pollen grain. Microspore nothing but the pollen grain. How they are to develop the microspore we are to discuss. After the we see here this diagram. So during the development of the microsporangium, the anther is in the first as the homogeneous mass of the meristematic cell belongs in the cross section of the surrounding by the epidermis. Here we can see the it then became the more or less four lobes and four longitudinal rows of the archisporial cells are the differentiated. The archisporial cells are marked off from the surrounding cells by their more or deeply stained cytoplasm and consequent nucleus approach. Okay. There may be only one such is the archisporial cells in each of the four lobes. Four lobes. So that is the archisporial cells are there. So there may be only one archisporial cell in each lobe, as in the Boerhaavia. Only one are there in the Boerhaavia. There are many formed in the Ophiophagous. Ophiophagous here having the archisporial different four or more archisporial cells. They are the longitudinally, and also there may be one or many are there. And each archisporial cells now cut off a primary parietal cells towards the epidermis as a primary sporogenous on the inner side. The parietal cells now is divided and by periclinal and anticlinal wall give rise to the several layers of the forming the wall of the anther. And the sporogenous cells are usually divided a few times due to the number of microspores or pollen mother cells. The innermost layer of the wall cells is directly abutting on the sporogenous tissues form the tapetum, which are the nuclear tissues nourishing the development of the microspore. We know that. So these wall cells are just below the epidermis from the endothelium, which are later lose the cell connection and usually became a fibrous and form the dry coat of the mature and which are the epidermis became a rather inconspicuous, inconspicuous character. So the between the tapetum and endothelium, there are the one there is the another special layer is that is the middle layer. These middle layers are the tapetum are usually crushed by the time actually meiosis occur in the sporogenous tissue. So during the microsporogenesis, the nucleus of the each microspore mother cell undergoes to meiosis, ultimately give us to four haploids. So these microsporogenous mother cells undergoes to the meiosis and to produce the four haploids. Four haploid cells are formed. So these four nuclei are arranged tetroidally. Tetrad condition. They are arranged in the tetrad condition. Okay. So here you can see the a tetrad-like structure. This one. Okay. So tetrad condition is there. Okay. So these the four nuclei are arranged in tetrad condition and are so inverted with the cell wall. So many variations are known for the typical pattern of the meiosis. In May, the walls are formed across the dia. So they are now the microspores or pollen grains. They are dried up. They became a powdery, while the tapetum became a observed. The anther now became a dry structure. The portion wall between the sporogenous sporangia are usually destroyed, and the microspore are soon liberated by the dehiscence of the anther. The tapetal cells are often became a multinucleate and play a great part in the nutrition of the pollen grains. Sometimes they are developed a plasmodium after degeneration and play the part of the development of the exine of the pollen grain. Even the part of the sporogenous tissues may be break down and serve for the nutrition instead of the development of the spores. While the pollens are dry and powdery in the most of flowers, and peculiar conditions are often led with the in anona. The spores are tetrad, never separate, but they are formed as the compound pollen grains, that's the pollinia. In the Mimosacea family, then most of they are to aggregate these pollen grains about 8 to 64 pollen grains are aggregated there in from the gynoecium of the calotropis and gynostigium of the orchid. So they are called as the pollinium. So all pollen grains are together to form a mass of the pollinium. That is the 4 to 84 around in number. So each pollen is provided with a stalk called cadicle and sticky base cells are disc or corpuscular. So the pollen or microspore is very minute structure is there about very It is the new unicellular and usually round, although maybe oval and pyramidal polyhedral in structure. Different structures are there. It is provided with the two coats are there, two layers are there. That is the coats are there. Nothing but the inner, that is the delicate cellulose layer that we call as the inner intine. So intine is there, and outer, which is very tough and cutinized layer, that is called as the exine. The pollen grain is made up of these two layers. The inner one is the cellulose contained, and the outer one is the cutinized material. So these exine is often the sculpture are provided with the spines and warts and etc. Some are the more. Okay. And So here, the milk gametophyte development, let's see this one. The nucleus of the microspore belongs to the is divide very soon after it is formed. Its the division is usually quicker in the warmer climate than in the color regions. Such divisions may begin even before the microspore dissociate from the tetrad condition. When the pollen is lobed on the stigma, usually the nucleus has already divided. The microspore cells divides into two very equal cells, which are the one is the smaller, the attached to the wall. The latter soon lost the contact with the wall and became the like a form the lens shaped with the generative cells floating the free in the cytoplasm of the a large vegetative tube cell or tube cell. It is then in the binucleate as the pollen germinate and the pollen tube come out to the genspore, the vegetative nucleus precedes the generative cell. The generative cells soon is divided mitotically to form the two male gametes, and this act as the spermatogenesis, they are known as the spermatogenesis, as the male gametophyte of the angiosperms. The equivalent to the ciliated sperms of the lower groups of the plant. The gametes nucleus are enveloped in cytoplasmic sheath. This is forming the gamete cells. At the latter stage of the two male gamete cells are seen to follow the vegetative nucleus. So soon these vegetative nucleus is important being a mere vegetational organ and disappear. So as the pollen in the microspore, the content with the pollen and pollen tube formed by the germination of the microspore in the male gametophyte. So while these male gametophyte does usually develop after the pollen that has the germinated in the instance where it developed with the pollen before the pollen tube are formed. So these trinucleate pollens in the two pollen tube nucleus and two gamete cells are observed in such some cases. Okay, so here